Okay, good morning, everyone. Good afternoon or good evening, wherever you are. I'm Tina Park from Harvard T. Chan School of Public Health. I'm delighted to serve as a moderator of this scientific symposium. It is entitled Lung Epithelial Damage and Repair from Cell Death to Lung Tissue Regeneration. We welcome each of you to join us this one of the most exciting symposiums in this year's ATS conference. Thanks, Dr. Powles, for putting together this exciting symposium. Today, we'll have three 20-minute talks. Each of you will be followed by a 5-minute discussion. Please submit your questions to the chat box during the talk. Then at the end of each talk, moderators will ask questions on behalf of you. Prior to three talks, we'll give you a brief overview of the symposium to share what this session is about. It's my pleasure to introduce my co-moderator, Dr. Mamita Ghosh, who will give you an introduction to the symposium. Now I'll hand it over to Dr. Ghosh. Mamita, please take it away.
Good morning and welcome. So in this session, in next hour and a half, we are going to focus on one of the major lung disease that we study is chronic obstructive pulmonary disease, or commonly known as COPD. And in the developed world, cigarette smoking is the major etiology for COPD, whereas in other part of the world that other environmental exposure like wood burning stove that leads to fine particles can also leads to COPD. So COPD is a heterogeneous disease and it can affect different parts of the lung. But for this particular session, we are going to focus on airway epithelium, which is really the fence, so the barrier that is protecting our lung from the external environment. And as the cigarette smoke leads to the damage to the epithelial cells, in next half hour and a half, we will learn they would die, and they die in different ways. So we'll learn about different modalities of death. And some of this death could be immunogenic, leads to a vicious cycle of inflammation. The questions are can we intervene? Are these death could be different modalities of death could be used as biomarker for who can get COPD and who will not get COPD, and is there a therapeutic intervention? So without further delay, we have three excellent talks, and I would introduce our first speaker, Dr. Simon Pauwels from University of Groningen in Netherlands, and he's going to talk about his work on the role of dams in epithelial damage and repair. So Simon, please take it away.
Thank you, Mamita, for the great introduction. It's also great to see that almost 60 people have already logged onto this session. Thank you all for that. My name is Simon Pauwels. I'm from Groningen in the UMCG in the Netherlands. And today I will tell you something about the role of dams in epithelial damage and repair. So I have nothing to disclose. So as most of you are probably aware, COPD is a very severe and progressive lung disease that is characterized by both chronic bronchitis, so the chronic inflammation of the upper airways, and you have mucus hypersecretion, as well as destruction of the alveolar tissue, so emphysema. And this both causes the patient to have troubles with normal breathing. So COPD is caused by a combination of environmental exposures, so long-term smoke exposure, but also exhaust fumes and or air pollution, and in combination with a genetic predisposition, so genetic susceptibility, because there are a lot of smokers or occupational people with occupational exposures that do not develop COPD. So it's basically the combination of genetic susceptibility with a long-term exposure to toxicants that causes COPD. So that's a bit better explained in this graph here. You see that I see the lung function decline over age, and all individuals from the age of approximately 25 years onwards, you will see a decline in lung function. But before they will reach the first threshold where clinical symptoms of COPD will arise, healthy individuals will die before they get symptoms. But now when you are genetically susceptible for the development of COPD, you become the green line, and your progression in the decline in lung function will go much faster. And then on top of that, on top of your genetic susceptibility, if you have a lifelong exposure to toxic gases like cigarette smoke, then you will become the red line. And then all of a sudden, at the age of like 55 or 60, you'll develop symptoms that coincide with COPD. And so far, treatments for COPD or that are known that can decline or decrease the decline in lung function, but they cannot revert you back above the threshold of symptoms. So they cannot cure the disease. So that displays that there is an urgent need for regenerative medicine that actually restore lung function above the threshold of clinical symptoms. So my research focuses on these 4 questions. So which molecules and pathways are involved in the inflammatory reaction which we see in COPD patients? And then can we use these pathways and molecules to stop the airway inflammation that we see in COPD? Another question is that we have is why do some smokers or occupational exposure get sick while other people do not? So what is the role of genetic susceptibility in these processes? So my studies focus on one particular group of molecules, so that is DAMs, damage-associated molecular patterns. And these are endogenous danger signals that are released from damaged or dying cells that alarm the immune system for cellular damage or cell death. So basically, to alert the immune system, there's something bad going on. You have to become active and you have to clear the mesh. So normally, under physiological conditions, DAMs are inside the cell, hidden for the immune system, or inside the ECM, hidden for the immune system, and they have all kind of normal homeostatic or physiological functions. But when a cell dies, then the intracellular content is spilled into the extracellular space, and now all of a sudden, these DAMs are visible for the immune system. And when a cell dies in a regulatory fashion or in an apoptotic fashion, the DAMs are nicely packaged into apoptotic bodies, and these apoptotic bodies are phagocytosed, and you, in general, do not get an immune response. But when a cell dies in an unregulated fashion, in a mostly necrotic fashion, the cell just bursts open, and all the intracellular content is spilled all out in the extracellular space. And now these DAMs can bind to pattern recognition receptors on cells of the innate immune system, and these cells can then get activated and induce a pro-inflammatory response. By now, there are about 30 or 40 different DAMs known, and this is a wild mix of molecules that goes from DNA to RNA to mitochondrial proteins to heat shock proteins or HMGB1. But all of them have in common that once they are released from a cell, that they can activate pattern recognition receptors on cells of the innate immune system, and this way initiate a pro-inflammatory response. So that's why we built this hypothesis, where we say that chronic toxicant exposure to the airway epithelial layer induces epithelial damage to such an extent that these cells die in a necrotic way. So with this process of necrosis, then DAMs are released, and these DAMs can activate neighboring epithelial cells. So they can activate pattern recognition receptors, and these epithelial cells then get activated and produce pro-inflammatory cytokines like IL-6 and IL-8. And these cytokines, together with the DAMs itself, can activate and attract cells of both the innate and the adaptive immune system, and you get an inflammatory response like we see in the airways of COPD patients. So for the remainder of the talk, I will show you all the evidence which we have gathered in the past 10 years to support this theory. So I will show you 7 different steps. So we started very simple. It's a very simple experiment where we have BS2B cells, so that is a bronchoepithelial cell line, and we exposed it to several concentrations of cigarette smoke extract. And for those of you who do not know what CSE is or how it's prepared, so here you see a cigarette which is attached to with tubings to a pump, and the pump sucks away the smoke through the tubing into a tube full of growth medium. And now all the water-soluble components of the smoke are dissolved in the medium, and you can use that to expose cells. So we diluted this CSE into several concentrations ranging from 0 to 50%, and then exposed this to our cells. And then we measured the amount of necrotic and apoptotic cells using an XM5PI. And in gray, you see that almost no apoptosis is induced, but in black, you see that a large amount of cells become necrotic. And when you expose them to 30% CSE for 6 hours, then almost all the cells die in a necrotic way. So then you want to know if this also induces damp release, right? Because the theory is that the necrotic cell death induces damp release, which then can activate other cells. So we did a very similar experiment. We did the BS2B cells, and we exposed them to 30% CSE, because we shown that that induces necrotic cell death. And then we measured damp release in the supernatant. So here you see 3 different DAMPs. You see double-stranded DNA, HNDP1, and mitochondrial DNA. So there's 3 different groups of DAMPs, and you see that all 3 of them are significantly increased in the cells that were exposed to CSE. So then we made it a little bit more complicated. So again, we exposed our cells to 30% CSE. So then we have shown now that DAMPs are released. And then we take our DAMP-containing supernatant and spin it down to remove all cellular debris and all cells which are still present, and used it a conditioned media to stimulate fresh new BS2B cells, and then see if they become activated by measuring IL-6 and IL-8. The simple answer is here, yes, they become activated because both IL-6 and IL-8 go significantly up. So then you want to know if this process is also happening in an in vivo situation. So we did a mouse experiment where we exposed mice to cigarette smoke. So here in the front, you see 2 boxes with mice that are exposed to cigarette smoke. You see the cigarettes here in front being connected to 2 pumps, which pump the smoke into the boxes. And on the back here, you see 2 boxes with mice which are exposed to control air. And we exposed them for 5 days with an increasing number, starting with 1 and ending with 2 times 5 cigarettes per day. And we euthanized the mice 2, 6, and 18 hours after the final exposure on the 5th day. So then we looked at the amount of inflammation in the airways. We see an increasing amount of neutrophils up to 18 hours after the final exposure. So then we also want to measure DAMPs in the bowel fluid of these mice. So here you see S100A8 and double-stranded DNA, 2 well-known DAMPs. And you see a very similar pattern of DAMPs being increasingly released after the final exposure on the 5th day, with the most release of DAMPs 18 hours after the final exposure. So indeed, when you expose mice to cigarette smoke for 5 days, you get DAMP release in bowel fluids. So we were thinking, how can we now inhibit this process to limit the amount of inflammation that we see in mice upon cigarette smoke exposure? So necrosis is a very passive process. The cell just bursts open and everything is released. So it's really hard to inhibit that. But fortunately, there is also a regulated form of necrosis called necroptosis. And here there are indeed pathways like the RIP1 pathway is activated, and you can intervene with this cellular process using the chemical inhibitor necrostatin-1. So that's exactly what we did. Again, we have here our BS2B cells, and we exposed the BS2B cells to 30% CSE for 6 hours. And we measured cellular viability using MTS assay. And you see again that 30% CSE causes a drop in viability. But then if we pre-incubate the cells for 1 hour with necrostatin-1, then there is no drop in cellular viability anymore. So we can completely remove the decrease in cellular viability. And then also when we look at DAMP release in the graph on the right, you see that the 30% cigarette smoke extract induces double-stranded DNA release, as we saw before. But if we pre-incubate for 1 hour with necrostatin-1, then all the DAMP release is inhibited. So this shows us that most of the cell death which is occurring upon cigarette smoke extract exposure in our epithelial cells is caused by necroptosis, because we can actually prevent this. So we were, of course, curious if we can also use this in our in vivo model. So here you see, again, mice. In the first white balls, there are air-exposed mice. And then if you expose them to smoke, you get neutrophils in bowel fluid, and you get airway inflammation. And then we intraperitoneally injected these mice with necrostatin-1 on a daily basis. And the necrostatin-1 itself did not induce any inflammation or something. But then if we expose these to smoke, you see a significant reduction in the amount of airway inflammation. A large part of the airway inflammation induced by smoke can be inhibited using necrostatin-1. So this shows us that there is great potential for necrostatin-1 to reduce airway inflammation. In this case upon smoking, but maybe also upon other exposures. So as I've told you, we were interested in identifying what the role is of genetic susceptibility in this process. So what is the role of genetic susceptibility in the release of DAMs? So we did a very similar experiment, exposing mice to cigarette smoke for 5 days. But now we did not use one particular mouse strain, like the previous experiment were with BALB/c mice. But now we use 30 different inbred mice strains. And for all 30 strains, we have 8 mice being exposed to smoke and 8 mice being exposed to air. And then we determined the amount of double-stranded DNA release in ball fluid. And here you see in this graph the difference in double-stranded DNA levels between the smoke and the air-exposed mice. And you see a big difference between the different mice strains. Some are really susceptible for smoke and they have a large amount of double-stranded DNA being released, but others do not respond at all to cigarette smoke, and they are completely resistant towards smoke. And this difference in how they respond is caused by a difference in their genetic background. So we were interested in which genes are now involved in this double-stranded DNA release. So therefore, we did a haplotype association mapping analysis, basically looking at SNPs which associate with the levels of double-stranded DNA in ball fluid, and then investigate which genes are related to the SNPs. And this way, we identified 11 genes which are associated with the double-stranded DNA release upon cigarette smoke. And the most interesting one maybe is CFLAR, which encodes the protein C-flip, which is a regulatory protein determining whether a cell goes into apoptosis or into necroptosis. So it's basically determining whether a cell releases its DAMs or not. So we validated this response using measuring CFLAR expression in lung tissue of never smokers and active smokers. And you see that the expression of CFLAR is significantly lower in lung tissue of smokers, indicating that smokers are more susceptible to release their DAMs upon smoke exposure. So we took it one step further, and now we selected 2 mice strains from the previous 30 that we did. One being really susceptible for cigarette smoke-induced DAMP release, and the other being totally resistant. So we have in white the BALB/c by J being the really susceptible mice strain, and in gray you see the resistant DBA2 strain. And then we intranasally, so in the nose, injected these mice with a single dose of LL37. That is one specific DAMP that activates both toll-like receptor 4 and RATES, or a group of mitochondrial DAMPs. And you see that in the resistant mouse strain, there's nothing happening. The DAMPs have no effect. There's no inflammation induced. But in the susceptible BALB/c by J strain, you see a big induction of neutrophilia upon the injection of DAMPs. So this shows us that there is a correlation between the susceptibility for smoke-induced inflammation and for DAMP-induced inflammation. And then we investigated more specifically the role of RATES, because as you will hear in the final talk of this session, RATES is one of the most important DAMP receptors, and there's a lot of evidence showing that RATES is involved in the pathophysiology of COPD. So what we did in this study is, again, intranasally, so in the nose, injecting the mice with either BSA as a control or LL37 as a DAMP. And then in the wild-type mice, these are black 6 mice, the LL37 induces airway inflammation, both total cell count and neutrophils. When we use RATES knockout mice, then the LL37 does not induce any inflammatory reaction anymore. So basically showing that the airway inflammation induced by LL37 is not induced by is induced by RATES and not so much TLF4. Then we prepared precision cut lung slices from black 6 mice, and then ex vivo treated these mice with a RATES inhibitor called FPS71, and then investigated the alveolar size to get a measure for tissue damage, for emphysema. And when we ex vivo treated these slices with LL37, then you get an increase in the mean alveolar size. So that induces tissue damage. But then if we pre-incubate these slices with FPS71, the RATES inhibitor, you do not get any increase in tissue damage anymore. So also the induction of LL37 in tissue damage is almost completely dependent on RATES signaling. So finally, we were interested in how potent this DAMP inhibition actually is. So we again have the precision cut lung slices and investigated the mean alveolar size. And now we ex vivo treated these slices with elastase to induce a lot of tissue damage. So you see that 1 day after elastase treatment, there's a big destruction of the alveolar structure. And then 1 hour before and 1 hour after the elastase treatment, we incubated with the RAGE inhibitor FPS SETM1. And especially when we incubate after the treatment with elastase, then you see a big decrease in the amount of tissue destruction which is done by the elastase. So basically, inhibiting RAGE can inhibit a large part of the elastase-induced tissue damage, which is an important clue that RAGE actually is very important inducer of tissue damage. So here you see the scheme again that I also showed you in the beginning. And I hope that I now have shown you that cigarette smoke exposure actually induces necrotic cell death in epithelial cells, that with this process dams are released, and that these dams are able to activate other epithelial cells for them to induce pro-inflammatory cytokine release and airway inflammation. And that we can inhibit this process with necrostatin-1 or by using RAGE inhibitors. So these are very potent, also thinking about future therapeutic potential in COPD patients. And we have shown that this whole process is dependent on genetic susceptibility. So it's the combination of cell death and dams upon exposure with genetic susceptibility to induce airway inflammation on top of that. So I would like to thank the people listed on this slide. And I would like to thank all 78 of you for listening to this session. And please ask your questions in chat, so I will try to address them.
Great, very nice talk, great work. So the question is that, do you know in the mice if the dams are released by airway epithelial cells, alveolar epithelial cells, or immune cells, or is it all these cells?
Well, in mice, that is really hard to determine where the dam release is coming from. So we have to refer back a little bit to the in vitro experiments that we performed. So we exposed different cell types to cigarette smoke extract to see where most of the damp release is coming from. And we saw that especially epithelial cells and neutrophils are very prone to release dams upon cigarette smoke exposure. So we think that epithelial cells, being the first cells to encounter the inhaled smoke, is the first trigger to release the dams and to initiate the inflammatory response. But after that, when you have the immune reaction has started, then also the neutrophils can contribute to the ongoing inflammatory status by also releasing dams upon exposure and keeping the inflammatory reaction ongoing.
Effect of RAGE inhibitor in preventing direct airway damage by elastase was very impressive. Have you looked directly to see if the inhibitor has any elastase inhibitor function?
No, not particularly. No, we did not look into that. But I do know that there are also other studies of others, and I'm not sure if Timothy Perkins will show that, but that show that also RAGE knockout mice are less prone to induced for elastase-induced tissue damage, and also for cigarette smoke-induced tissue damage. It cannot be a direct effect of the RAGE inhibitor acting on elastase itself. It probably more a cellular response, because it's also RAGE knockout mice are also less prone to induce cigarette smoke-induced tissue damage.
Have you monitored carbon monoxide, carbon dioxide, and oxygen levels in your mouse smoking exposure chamber?
Yeah, no, we have not done that. We have not measured that and have not assessed that at all.
Can you discuss the comparison of the in vivo signal to DAMs versus PAMs like TLR4 or 7?
Of course, that will be very similar, because it's the same responses being activated, right? Both the PAMs and the DAMs activate the same receptor, so you get a very similar process. So probably it will be a very similar effect that you will see, but you won't have an airway inflammation going on every day. Well, people who smoke or have an occupational exposure, they will have the same exposure every day. So they will have the chronic insults towards their epithelial cells. So you will have chronic damp release. And I think that's an important trigger for that to contribute to COPD, the chronicness of the response.
So do you know why necroptosis is the primary form of cell death in response to cigarette smoke extract? And what component of cigarette smoke extract causes the cell death, which probably would be impossible to say.
Yeah, that is it will be very interesting to investigate which component of smoke actually activates this pathways. But at this point, I have to say that we have not looked into different components of cigarette smoke. Why necroptosis is so important in comparison to other cell types? I hope that maybe Suzanne Klonen, the next speaker, will give a better overview on the different types of cell death and how they may contribute to these types of responses. So in this case, we only investigated necroptosis, and we saw that very strong and striking effects. But there are, of course, also other cell type modalities which are very important, which may be very important.
Okay, I'm delighted to introduce Dr. Suzanne Klonen as the second speaker. Dr. Klonen is an associate professor of respiratory biochemistry in Trinity College Dublin in Ireland, and she is also affiliated with the Cornell University. I think she's been very productive because she has 2 labs up and running. I'm jealous about that. And there are different ways of cell death occurs in either in a programmed cell death or non-programmed cell death. And today, Dr. Klonen will discuss different types of cell death and how epithelial cell death could contribute to inflammatory lung disease. Dr. Klonen, please take it over.
Thank you, Dr. Park. So thank you for the organizers, Simon, for the invitation to give this talk today. This is a really difficult task to talk about. It's a vast body of literature. So I'm going to do my best to try and give a sort of an overview of the key papers in the last few years and talk a little bit about our work and try and maybe address some of the concepts that, we've just been asked about in the chat, but really we're not really there yet with this level of research, so we probably can't answer all of them. But I think we'll get it, initiate a good discussion for the end of this session to talk about ways we can do more. I have no financial disclosures. So in the last few years, we've in the last decade, there's been an absolute burst of knowledge with regard to cell death modalities and types of cell death and their role in disease and in biology in general. I guess this is an oversimplified schema, but we could separate cell death into either programmed or unprogrammed. And for a very long time, we thought that just programmed cell death involved apoptosis and unprogrammed was necrosis. But in the last few years, as I mentioned, there have been a number of other types of cell death pathways, including necroptosis, pyroptosis, ferroptosis, and netosis, that all play an important role, particularly in lung biology and disease. Intrinsic apoptosis and extrinsic apoptosis are probably the most well-known and most well-defined programmed cell death pathways that we know about. They involve distinct receptor-mediated or intracellular-mediated or driven effects that have a downstream signaling pathways that result in the formation of an apoptosome and lead to membrane apoptotic blebbing, where the cell tries to sort of maintain its structural contents, releasing them into the extracellular space before its final demise. However, there are many, many other types of non-apoptotic cell death, and some of them I can't even pronounce. So Simon has talked about necroptosis, which again is a rapidly emerging field. Every few months there are new pathways, new molecules that are important in necroptosis. We have pyroptosis, which involves more immune cell driven cellular mechanisms of death. We have ferroptosis, which is a lipid oxidative mediated stress form of programmed cell death. Paranthrosis, hope I'm pronouncing that correctly. Entotic cell death. We have netotic cell death, which again is involves the secretion of nets from neutrophils. We have lysosomal dependent cell death, autophagy dependent cell death, alkylapoptosis and oxyapoptosis. And these are just from 2019, and I'm sure there are more since. But these are varied and in their mechanisms to result in how the cell eventually dies, and quite a variety of these cell death pathways are involved in COPD, which I'll talk a little bit about later. So in the last, I guess, as I mentioned, the last 20 years or so, there was the most of these cell death pathways have been documented. You can see the timeline here of when these cell death pathways were discovered and their progression. As I mentioned, apoptosis was the first in 1972 to be described. And we have netosis, necroptosis, which is described in 2015 by Juan's group, and ferroptosis more recently in 2012. You can also see that in the last 20 years, there's been a burst in the number of publications related to cell death and programmed cell death pathways in the literature, striking its importance in understanding cell biology and also as a therapeutic target for many different diseases. So today I'm just focusing on COPD. Simon obviously gave a lovely introduction into the background of COPD and the combining factors that we believe are important in the pathogenesis of COPD, and genetic susceptibility is something that my lab works on. But to just give you an overview of the statistics of COPD, for those of you who don't know, it is the third leading cause of death worldwide. It affects over 16 million people in the US. However, there are thought to be around 12 million people who are undiagnosed with COPD, and it costs healthcare systems billions and billions of dollars a year. So in terms of the cells of the lung, today my task was to talk about epithelial cells specifically. And so what you will see from what I could find in the literature, the majority of cell death pathways that have been described in the context of COPD have involved airway epithelial cells, including ciliated epithelial cells, basal cells, goblet cells, and various other cell types. There have been a few papers documenting the role of these cell death pathways in the alveoli, specifically in the type 2 alveolar epithelial cell or the type 2 pneumocyte. But again, we're very limited. Majority of studies do not distinguish between the types of epithelial cells that they are discussing and just usually use a blanket description of a lung epithelial cell. And as you know, and you can see from this picture, lung epithelial cells vary dramatically in their appearance, their metabolism, their behavior, their secretory pattern. And so we have to really place importance on what cell types we believe. And I think that was one of the questions, but I think that's a really, really important question that we need to address as a field. So with regard to the cell death pathways that I just mentioned in COPD, I could talk for hours on each of these key papers, but we know that apoptosis has been mentioned many, many times in the pathogenesis of COPD in epithelial cells. But necroptosis, which is emerging, and the last, I guess, 2014 was the first paper to really describe it in COPD. And then these are some of the other key papers that have built on. This includes Simon's work and others who've shown that necroptosis plays a role in the release of DAMPs in COPD. And pyroptosis has also been described to play a role in COPD, specifically in epithelial cells, as has ferroptosis in 2019, paranthosis as well, that in 2019 as well. Nothing related to entotic cell death I could find. In terms of netotic cell death, which obviously is mostly specific to neutrophils, there have been a number of papers in COPD, but also lysosomal cell death as well as autophagy-dependent cell death have been really vastly described in the COPD literature with regard to epithelial cells specifically. So it's hard to address the question of how damage by various cell death pathways lead to specific routes of inflammation or particular pathways, because nobody's really studied these in depth. I mean, we're only in the last 5 to 10 years really have we began to use targeted cell approaches to look at these molecules and these pathways in the lung, in mice especially, and in humans obviously this is extremely hard to do. But there, have been a number of nice reviews discussing the role of the effects mainly of cigarette smoke on here and the epithelial cell, which to me looks like an airway epithelial, ciliated epithelial cell barrier, and how response to cigarette smoke will will induce the release of DAMPs from these epithelial cells, which will then lead to downstream immune signaling pathways, including the activation of macrophages, various other innate immune cells, and as well as the adaptive arm of the immune system, which ultimately results in the development of emphysema or mucus hypersecretion and small airways disease, and also in fibrosis. And so there have been a number of papers, but again, not really, no real direct linking mechanistic papers that link a specific type of alveolar epithelial cell with a direct link to some of these mechanisms. There have been many people who have touched on this, but not in really in depth. Again, there's this concept, apologies, this slide didn't come up as nice as I had hoped, but there's another concept of what is the effect on cell death on bystander cells in the airways or in the alveoli, and how these damps or other secretory molecules have an effect on, for example, neighboring macrophages or infiltrating neutrophils, and how the microenvironment then of the lung contributes to their activation. So there have been a number of signaling pathways. For example, here apoptosis can induce a number of responses in a macrophage through the TAM receptor, regulating particular types of cytokine responses. And similarly with necroptosis, it's thought that these necroptotic secretory phenotypes will cause, again, a specific signature in a bystanding macrophage. But again, with regard to COPD, this has yet to be defined. So in my mind, the gaps in our knowledge are precisely what one of the questions was: what epithelial cells produce what damps, and how selective this is. Is there a localized or systemic effect of a damp once it's released? How can we target these damps and use them for targeted cell therapies in COPD? And I think in the literature, we really need to really distinguish when we're writing up our work and looking at our work, is this an alveolar or an airway specific phenotype that we believe is important in COPD? And again, the literature is quite gray in terms of, most people will talk just about the blanket epithelial cell, and I think we need to start focusing in on specific epithelial cell types and what their role and what specific damps they play. So I'm going to just use an example of a well-known, I guess, signaling pathway in epithelial cells, or that we have shown we've worked a little bit on, but again, not much. I haven't worked in this area for a number of years, but I'm going to talk a little bit about the mitochondria. For those of you who know me, you're not going to be surprised, but I just thought it was a good example of a damage associated, well, in my mind, it is a regulator of damps inside the cell and outside the cell. And I think it has been shown to contribute to nearly every programmed cell death pathway that I've mentioned earlier. So I think it's an important thing to discuss today. I also, again, just to rehome my advice in terms of like what type of epithelial cell you believe is important for your work and what you should study based on what molecule you're studying. For example, the mitochondria have varying structural and functional components in very different epithelial cell types. So we know in the ciliated epithelial cell, the mitochondria plays a role in providing energy for cilia beating and for highly energetic function of this cell. And whereas we know in type 2 epithelial cells, it most likely plays a role in the secretion of surfactant or lipid regeneration and lipid biology. And so you really have to keep in mind, again, the cell type that you're looking at with regard to whether it's mitochondria or whether it's regard to your specific damp that you're interested in. So just again to highlight here briefly that the mitochondria is present and it plays a role in nearly all of these programmed cell death pathways, including autophagy, apoptosis, paroptosis, netosis, ferroptosis again. So they play an important central mediator, and I think of a lot of these cell death pathways. And they specifically have an array of damps that they can secrete in response to injury. And these damps can act intracellularly or extracellularly. So for example, cardiolipin, which is a mitochondrial specific lipid only found in the mitochondrial inner membrane, once that's secreted from the mitochondria and it's found in other places, such as the cytosol or the extracellular space, it can act as a damage associated molecular pattern, as can calcium, iron, reactive oxygen species also is a well-defined damp that we know plays a role intracellularly and extracellularly. And these damps will ultimately lead to the activation of inflammatory cascades. These damps in mitochondria have been implicated in many other lung diseases, not only in COPD, but in asthma, pulmonary hypertension, lung cancer, lung infection. And so there's another body of literature that describes the role of these damps and their role in lung biology and disease. So again, to focus on the role of COPD and in the epithelial cells specifically, we had shown previously that mitophagy, or the selective removal of mitochondria by autophagy, plays an important role in the mechanisms by which cigarette smoke induces cell death in an epithelial cell. We believe that the activation of necroptosis is involved in this pathway, and It's an important downstream signaling effect of excessive mitophagy. And one of the proposals of this work was that mitophagy would ultimately regulate the amount of mitochondrial DNA, which is a mitochondrial damp secreted by an epithelial cell, and many others have shown that mitochondrial DNA can be secreted by an epithelial cell in response to smoke. And so when mitochondrial DNA is secreted outside the mitochondria, it can activate, in the cytosol, it can activate the NALP3 inflammasome. So it can, in addition to reactive oxygen species and engagement by TAL signaling pathways, as well as a number of other signaling primers, you get activation of this IL-1 beta driven and NALP3 inflammasome response. And mitochondrial DNA is pinnacle to this, this activation and this response. And so others have shown that basically mitochondrial DNA, as well as I mentioned earlier, cardiolipin, but if you look specifically in the top right, mitochondrial DNA is thought to be involved in the activation of neutrophils, for example, and leading to multi-organ failure and SIRS, and also plays a role in many other lung diseases and lung infection. We still don't know much about it in COPD, how it plays a role in COPD. And so a close colleague of mine, Kiichi Nakahira, in 2013 had carried out this really nice study where he showed that increased mitochondrial DNA levels were associated with ICU mortality. In other words, the higher the amount of extracellular mitochondrial DNA in these patients' bloodstreams correlated with morbidity in these patients. So they had a high mitochondrial DNA signature in the blood, and this associated with their demise in the ICU, suggesting it as a damp, this signaling cascade and overactivation of the signaling cascade and plays a role in critical care illness. So we decided to evaluate if mitochondrial plasma, mitochondrial DNA extracellularly could play a role in COPD. And so we worked closely with the Spiromics cohort and measured extracellular mitochondrial DNA in 700 plasma samples randomly selected from this cohort, consisting of a mixture of never smokers, smokers without airflow obstruction, and then those with mild, moderate, or severe COPD. And what we found was there were very subtle differences in the amount of extracellular mitochondrial DNA present in the bloodstreams of these individuals, specifically in mild to moderate COPD. In the mild to moderate COPD cohort, there was a slight bump in mitochondrial DNA compared to healthy smokers. However, this declined with severity of COPD, going back down again, suggesting maybe a sort of biphasic response. And I'll talk about that in a little bit, what I mean by that. When we looked at plasma mitochondrial DNA and clinical outcomes in this study, we didn't find many associations, in fact. Mitochondrial DNA did not associate with FEV1 % predicted, 6-minute walk, SGRQ, CAT, all of these parameters, suggesting that it may not really play a role in the development or the overall disease progression in COPD. We also looked at extracellular urinary mitochondrial DNA and whether this was altered in COPD. And the reason why we did this was because there is some evidence to suggest that chronic kidney disease is more prevalent in individuals with COPD, and perhaps a kidney injury marker would also give us some insight as to a nice new COPD biomarker. So we looked at mitochondrial DNA, and we've previously shown also that mitochondria are damaged in the kidneys of smoked mice, and so we wanted to see if mitochondrial DNA could play a role and act as a biomarker in the urine. And so we did the same type of study, 483 samples. We measured mitochondrial DNA in the urine in a select group of these individuals. And we found that while there was no overall change in the levels of mitochondrial DNA between the subgroups in this study, there was a strong association between the amount of mitochondrial DNA and percent FEV1 predicted. But most interestingly, we found that in subgroup analysis, particularly looking at females, we found that mitochondrial DNA levels were higher in females. Urine mitochondrial DNA was higher in females. We did not see this in the plasma mitochondrial DNA. In the urine mitochondrial DNA, they were much higher in females. And in particular, females, the more mitochondrial DNA they had in their urine, the worse outcomes they had on some of these clinical outcomes, including 6-minute walk. And so there was some suggestion that by subgroup analysis that mitochondrial DNA in the urine could play a role and tell us a little bit about the physiology of the disease. So when I talked about the plasma mitochondrial DNA levels that we've seen, we think that in maybe an early mild moderate disease, there's a rise in mitochondrial DNA levels, which may associate with a more exacerbating active disease mechanism, whereby you get the release of mitochondrial DNA by some of these cells, and you see like an increase in the bloodstream. However, later in disease, there may be what I would call a mitochondrial exhaustion. So you see the depletion of mitochondrial DNA systemically, and you see this loss of mitochondrial DNA in severe emphysema patients. I know Simon's group have shown before that mitochondrial DNA can be secreted by cigarette smoke extract, both in vitro and in vivo, and this sort of fits because our animal models at this time points would be more indicative of a earlier COPD phenotype, and so cigarette smoke may induce mitochondrial DNA at this early time point. However, later on, when you see this sort of cachectic patient, or a patient with severe muscle wasting or severe emphysema, which many people have shown here. This is a nice study in Scientific Reports in 2018 showing that COPD patients' muscle biopsies show less expression of mitochondrial DNA copy number overall, as well as specific components of the electron transport chain that are downregulated, showing that they have just less mitochondria in general. So this is maybe why we're seeing less mitochondrial DNA floating around extracellularly in the severe COPD. And so what can damp mitochondrial DNA tell us about COPD? As a damp, as I mentioned earlier, we still don't know the cell type of where this could be coming from, and if there is a cell type specific function of damps in COPD in general. And with regard to plasma mitochondrial DNA, is this biphasic response? Is this telling us something about the physiology of the disease? What are the compartment differences when we look at these damps? You know, we look at plasma and urine. Should we look at BALF? I mean, many others have shown that mitochondrial DNA is upregulated in the BALF of patients with idiopathic pulmonary fibrosis. So should we look at BALF as well in COPD? What are the sex differences when we're looking at our studies? In these subgroup analysis, mitochondrial DNA may play a role, and damps may be sex predictive in a disease model. And again, supporting these all support a mechanistic role for mitochondria in COPD. And then, so in the outstanding questions in this field is, what is the source of mitochondrial DNA, or what is the source of these damps? I think that's a really outstanding question that we need to address as a field. With regard to mitochondrial DNA, is this related to the cachexia and loss of the muscles, muscle of severe COPD? Is the sequence of the mitochondrial DNA important, as with other damps and other? I mean, that's something I wanted to ask Simon in this double-stranded DNA, is have they looked at sequences and how specific the sequence might be for eliciting certain types of inflammatory pathways? And is this a marker? Probably not a good marker, or is it more a pathogenic mediator that can tell us something about disease? And I think the latter is probably what I would think is the most important. And so with that, I'll end. I hope I'm within time. I think so. I'd just like to thank Dr. Choi for some of the slides. He helped with giving a little bit of background of these cell death pathways, and to my funding agencies. And again, I don't think we can really answer exactly where these damps are coming from, but I think the field is moving in that direction.
It was a great summary of cell death mechanism, and it was very nice to show that the mitochondrial DNA is implicated in COPD. So while we have many questions in the chat box, do you know if it is differential release of mitochondria, or just both the nuclear and mitochondrial DNA?
Don't know, Marta. Don't know. I mean, if you have a lot of these cell death pathways, you would assume it's both, but I don't know. Even we haven't looked at that mechanistically.
Have you looked into mitochondria DNA in bowel fluid?
Yes, we've tried to measure it in the bowel fluid of these patients and really haven't been able to get a good yield. We tried, I think, 20, 30 patients who couldn't detect it very well enough to make any conclusions.
So you were only able to measure in the urine, not in BAL?
And in the plasma, yeah. The BAL we tried just a few samples. We could probably go back to do it, but yeah, it wasn't dramatically different, or it was just wasn't reliably detected. You know, we couldn't make a
I
firm
see. I'm just kind of curious. Do you know if this mitochondrial DNA are included in the extracellular vesicles?
We tried to do that before as well. Well, this is a lot of Dr. Choi's work. He had a postdoc who tried to do that in the past, which, you know, it's hard to get extracellular vesicles from plasma, really, really hard. Maybe, but nothing firm. Again, hard to make that conclusion. I imagine it probably is, especially in a disease setting. Maybe in acute or critical illness, you'd be able to see more because I think there's more of it floating around. But in the context of COPD, I don't think we really see, you know, that much of it. Unless maybe you choose like a really severe exacerbating cohort that are, you know, have active infection, maybe you might be able to get a better chance. I imagine it probably is there. It would make sense.
Do you think that the decrease in mitochondrial DNA numbers in severe COPD as compared to intermediate group is due to the loss of cells or a drop in mitochondrial biogenesis of the remaining cells?
Probably a bit of both. I know it's hard to study this, right? Especially with our experimental models we have, because in my experience with cigarette smoke extract and in vivo cigarette smoke exposures, there's this sort of early, what we call a hormesis effect on the mitochondria. So hormesis is basically a little bit of bad can't do you harm. So basically it's, if you stress out a cell, the cell will actually activate biogenesis, mitochondrial biogenesis. You see increased respiration of the mitochondria in that cell, as if they're trying to compensate, you know, they're trying to build up their mitochondria to deal with this stress. And we see that experimentally. If you smoke mice early on in COPD, you basically see an increase in mitochondria, and then there's suddenly a drop off, right? Which there's this rapid decline, and the longer you smoke, the more cigarette smoke extract. I mean, we know these experimental models that we use, everyone makes cigarette smoke extract differently. Everyone uses different toxicity, different percentages of cigarette smoke and longer time points. You can see whatever you really, with mitochondria, you can see any sort of stage. With regard to the muscle, I think, I would, you know, there's definitely, I think cell loss will be important, right? And that's something that we always need to look at the nuclear DNA to kind of get an insight on that as well, right? Because you have the nuclear to mitochondria ratio will give you an indication of how many mitochondria there are per cell. But I do think there's less per cell. And I think it's probably maybe a genetic component to that as well. I think there's an interesting side angle of this concept of mitochondrial heteroplasmy playing a role in some of these diseases. And, you know, if your sequence is different or you have a higher level of heteroplasmy to begin with, maybe you lose mitochondria faster. They're all kind of outstanding questions.
Release of extracellular vesicles or exosomes. I think probably we asked that question because the Louis Ortiz at the University of Pittsburgh showed that mitochondrial DNA has been detected in the extracellular vesicles or exosomes.
Yeah, I think in the lung as well, Yahar Batra's group have shown that the mitochondria can be transferred from cell to cell, and maybe in extracellular vesicles. Like, it's just technically really hard to measure in a mouse, and even in humans, to get enough plasma to do that, especially in a disease like COPD. But it's not to say it's not important.
Does the increase in one kind of DAMP have an influence on others? For example, are there other DAMPs increase in the severe COPD patients?
Yeah, why not? I mean, there has to be a synergistic effect by some of these receptors. I mean, it would make sense that you have multiple cytokines or DAMPs, you know, activating a particular cell type, like the macrophage, for example. You know, there must be more than one alveolar epithelial cell DAMP that activates a macrophage. So I wouldn't, why not mitochondrial DNA being synergistic with something? As you see with the NALB3 inflammasome, in order to activate the inflammasome, you need a number of signals. You can't just activate it with one thing, right? It needs to be both intracellular mitochondrial DNA. You need to have a Toll-like receptor engagement as well in order for that to all work. So perhaps in the setting of certain immune cells, you need mitochondrial DNA as a primer for something. And then another signal that acts to really control. This is highly complex, right? So it could just be different combinations of different DAMPs could elicit different responses in different immune cells.
Okay, great. Thanks a lot, Dr. Cullinan, for all your
Thank you.
answers and responses. And I really enjoyed your talk, and I'm sure others did as well.
Thank you.
Good luck for the rest of your the sessions and meetings.
Thank you. Thank you, Dr. Park.
Then we are now in the last talk of this session, and we have Dr. Timothy Parkins from Department of Pathology, University of Pittsburgh, and he is going to talk about RAGE, DAMP receptor with therapeutic potential in COPD. So here you go, Dr. Parkins.
Thank you very much for the introduction. And thank you to the organizers for putting on this great meeting and giving me the opportunity to be here today to talk to you about RAGE, a DAMP receptor with therapeutic potential in obstructive lung disease. And I have nothing to declare. So RAGE is the receptor for advanced glycation end products, first discovered for its ability to bind advanced glycation end products and was associated with cardiovascular complications and diabetes. However, it binds a multitude of different ligands. It is a DAMP receptor. Its expression is highest in the lungs, most notably in the type 1 alveolar epithelium. However, it's also able to bind type 4 collagen as well as beta-2 integrins, and it's mostly associated with promoting sustained inflammation in various conditions. RAGE also exists as a soluble form, which is created by endogenous secretion of alternative splicing or cleavage by matrix metalloproteases, and this acts as a decoy receptor to negatively regulate RAGE signaling. There's a number of different SNPs in the RAGE gene in various different conditions. However, this one in particular is associated with various chronic inflammatory conditions. It results in a glycine to serine substitution in the ligand binding domain, leading to increased signaling capacity. It's also associated with decreased sRAGE levels, as well as variation in lung function. RAGE signaling is highly complex, but what we do know is that upon ligand binding, you activate the receptor. Although it has no intrinsic kinase activity, it's known to signal via recruitment of diaphanous 1 to the plasma membrane, activating various kinase signaling cascades, including the MAP kinases and NF-kappa B, and this results in the induction of various inflammatory mediators as well as the RAGE gene itself. In the environment, we're all exposed to various noxious particles and gases, cigarette smoke, particulate matter, as well as allergens like house dust mite antigen. And in those susceptible individuals, this leads to persistent inflammation, aberrant mucus production, and airway obstruction, ultimately leading to airflow limitation. And what I'm going to talk about today is a great deal of data that suggests that RAGE is a key contributor to the development of obstructive lung disease. I'm going to go over some of the major stuff with COPD and emphysema, as well as some recent data, which I found very intriguing. And then I'm also going to talk about a critical role that our laboratory and others have found for RAGE in development of asthma. So to start with RAGE and COPD, initial studies found that in subjects with COPD, you had an increase in RAGE expression in the airway epithelium, as well as the submucosal glands, and in the airway smooth muscle cells. Another recent study also showed increases in RAGE expression in peripheral lung tissue homogenate from subjects with COPD. So while RAGE expression is increased in the lungs, we also know a number of studies have shown RAGE ligands are also increased, including AGEs, HMGB1, S100, A12, for instance. On the other hand, studies have consistently shown that systemic sRAGE levels are decreased in subjects with COPD and emphysema. And similar studies have shown this to be consistent in idiopathic pulmonary fibrosis as well. However, it's unclear if this is cause or effect, whether this promotes increased RAGE signaling and inflammation, or if this is just due to damage to the alveolar epithelium, which is the major source of sRAGE. This SNP as well is also associated with decreased sRAGE levels. However, it's also shown to be involved in very natural variability in lung function in healthy individuals. However, examination of a pool of smokers with and without obstructive lung disease, it was shown that this SNP was more frequent in those without obstructive lung disease, suggesting it could be protective. So it's very unclear how this is involved in the development of COPD and emphysema. And as mentioned earlier, a great deal of studies from various investigators have shown that in multiple models of obstructive lung disease, including cigarette smoke exposure, elastase-induced emphysema, or particulate matter, RAGE knockout mice are pretty consistently protected from inflammation and the development of emphysema. And now I just want to touch on some recent data which we've seen, looking at the effects of smoking directly on how RAGE behaves. And this recent study showed that both RAGE and TLR4 were decreased gene expression in bronchial biopsies in both healthy and smokers with COPD. However, they noticed that there was a positive correlation of RAGE gene expression with neutrophilia and a negative correlation with lung function. Airway hyperresponsiveness is an important parameter assessed in various models of obstructive lung disease. And recently, the same group showed that acute cigarette smoke exposure induced airway resistance after methacholine challenge in wild type, as well as TLR4 knockouts. However, RAGE knockout mice were protected from this. In addition to that, they saw that RAGE knockout mice had reduced neutrophils. And another similar study using a different type of model showed that RAGE knockout mice were also protected from the development of airway resistance after exposure to World Trade Center particulate matter. This all suggesting that not only does RAGE contribute to airway inflammation, especially neutrophilia, it is also involved in regulating lung function. So what about smoking, the effects on S-RAGE levels? And a very interesting paper from Dr. Poles and his group recently showed that systemic S-RAGE levels were decreased in subjects with COPD from mild to severe compared to both young and older control subjects. And very interestingly, they assayed S-RAGE before smoking and then 2 hours afterward, and showed that there's a decrease in almost all subjects assayed after smoking. Very intriguingly, this decrease in circulating S-RAGE is very rapid, happening within 1 hour and persisting for up to 48 hours. Now, how this contributes to pathology is unclear, and if it does, and the mechanism of inducing RAGE decreases is unclear, but they do make a very important note or hypothesis that after smoking, subjects usually see a large influx of neutrophils into the bloodstream. Neutrophils express beta-2 integrins, and S-RAGE is able to bind this. So somehow it could be that this influx of neutrophils is reducing S-RAGE, but how this contributes to disease pathology is unclear. Another very important paper from back in 2012 showed that S-RAGE was deficient in the lungs of subjects with neutrophilic asthma or neutrophilic COPD compared to those without neutrophilia, showing like a very clear association of S-RAGE and neutrophilia. And these subjects most often have much more severe disease, especially those with neutrophilic asthma, and they tend to be resistant to conventional corticosteroid therapies. So what about RAGE and asthma? Now, our group and many others have shown that there is a pretty important role for RAGE in experimental models, and there's also a lot of data out there suggesting that RAGE ligands are increased and S-RAGE is reduced in asthma. So asthma is a term used to describe a number of different endotypes of asthma of variable severity, all with the common feature of reduced airflow, at least partially reversible airflow obstruction. In asthma, we see airway mucus metaplasia, increases in mucus hypersecretion, airway smooth muscle hypertrophy. You usually have peribronchial and perivascular inflammation, all resulting in obstruction of your airways. In terms of the endotypes of what we know the most about, I would say type 2 high asthma is the most well characterized. And in terms of mechanism, we do know that exposure to allergens or other insults causes epithelial damage and then release of epithelial damps such as interleukin 33, TSLP, and IL-25 being the most prominent three. And these promote the activation and interaction of both the adaptive and innate immunity, perpetuating robust type 2 inflammation, leading to development of asthma. And in recent years, the type 2 innate lymphoid cell, the ILC2, has emerged as a key player promoting production of massive amounts of type 2 cytokines, leading to tissue eosinophilia, tissue remodeling, bronchial hyperreactivity, and mucus metaplasia. So what about the role of RAGE here? I'm going to walk through a bunch of data from both our group and others, which I think will give you a very intriguing storyline that RAGE could be a key contributor here. So to start, it was first examined the effects of chronic house dust mite exposure on wild type and RAGE knockout mice. What we found was that compared to wild types, RAGE knockout mice were protected from allergen-induced features of asthma, including airway hyperresponsiveness. They're protected from eosinophilic inflammation in the airways. They lacked type 2 cytokine production in the lungs. And furthermore, they were protected from the development of mucus metaplasia in the airways. This very similar results have been seen by our group and others using other models of allergic airway disease, including ovalbumin, cockroach allergen-induced asthma, as well as the the fungal allergen Alternaria. We were also able to see that administration of exogenous S-RAGE was able to significantly reduce airway eosinophilia and inflammation in the airways after exposure to house dust mites. What about during the allergen sensitization phase? Now, this slide is going to confuse you, and that is definitely the point because it's very unclear what roles RAGE is here. An initial study from 2014 showed that RAGE was able to promote Th2 cell sensitization after mice were administered HDM and sensitized Th2 cells were then isolated from the lymph nodes and stimulated ex vivo, showing that wild-type mice produced large amounts of IL-13, whereas both RAGE and TLR4 knockouts were protected from this. In addition, the authors showed that there was significantly less dendritic cell trafficking or expanding in the lungs and lymph nodes, suggesting RAGE may be involved in antigen presentation sensitization. In line with this, we also saw that after exposure to Alternaria extract, wild-type mice have an increase in CD4 positive, IL-5, and IL-13 positive cells in the lungs, whereas the RAGE knockouts are not. And this is where it gets confusing because we know that after chronic house dust mite exposure, these mice don't develop features of asthma. However, you will see they produce normal amounts of IgE and they make HDM-specific IgE and IgG. So it's very curious that they don't develop features of asthma, but they seem to be clearly found the antigen, recognize the antigen, and have been sensitized. So this is something that definitely certainly warrants further research. So what about the DAMP signaling? And I think we should focus on IL-33 here and look at IL-33 signaling and the role of RAGE. And what we have seen was that RAGE was required for the accumulation of IL-33 in the lungs of mice after exposure to Alternaria extract. Similar results were seen with house dust mites, suggesting RAGE is required for IL-33 to be released and accumulate in the lungs. So what if we could bypass that and the lack of accumulation and just give recombinant IL-33 directly? And what we found was that RAGE knockout mice were further protected from recombinant IL-33 induced allergic airway disease, which is very curious because we found normal levels of ST2, the IL-33 receptor, in the lung. So it's unclear what the mechanism here is, but this is two steps already that RAGE is pretty important. So IL-33 signals to the ILC2s and induces their accumulation and production of type 2 cytokines mediating allergic airway disease. And what we found was that after exposure to either Alternaria extract or IL-33, we saw there was no accumulation in RAGE knockouts of ILC2s. However, if you give an intraperitoneal injection of IL-33, you get normal accumulation of ILC2s in the gut, suggesting RAGE's role in mediating IL-33 signaling is specific to the lungs, which is not surprising as there's quite a bit of expression in the lungs and very little in the gut. So we next thought, can we bypass the ILC2s and just give recombinant type 2 cytokines directly, assuming that we're going to see normal signaling? And very much to our surprise, we see again that RAGE knockout mice were protected from type 2 cytokine IL-13 induced mucus metaplasia markers, and you can see the pink mucus staining in the airways. In addition, they lacked production of eotaxins and CXCL1. They also lacked the mild neutrophilia and eosinophilia we saw in the airways. We also saw very similar results with IL-4. Let's look at the signaling. We know IL-13, IL-4 signal via the STAT6 pathway, binding and causing phosphorylation of STAT6 and inducing transcription. So we looked at STAT6 phosphorylation kinetics and found that in the very early time points, within the first hour post-stimulation, we had similar levels of phospho-STAT6, but by 8 hours, this was significantly reduced and almost not detectable at 24 hours. This was coupled with also an induction of STAT6 response genes only in wild-type mice, but not RAGE knockouts. So what we've seen here is that in RAGE expressing cells, we have sustained phosphorylation of STAT6, leading to transcription and ultimately mucus metaplasia and inflammation in the airways. Whereas RAGE knockout mice, we only see transient phosphorylation and the airways appear normal. So can we target this pharmacologically using a RAGE-specific antagonist? So we administer the RAGE antagonist that Sandra spoke about earlier, FPS-ZM1, at the time of stimulation with type 2 cytokines. And we were able to see that this was able to significantly reduce markers of mucus metaplasia, as well as neutrophils and eosinophils in the airways. So we also want to see, well, can we see this in a human cell line or a primary human cells differentiated air liquid interface as well? And we administered right before IL-13 stimulation, and we show again in a human primary human cell, we can block the induction of mucus markers as well as inflammatory markers. So what we know now is that RAGE is critical at multiple steps in the development of allergic airway disease. We know that RAGE is required for release of DAMPs, including HMGB1 and IL-33. We know RAGE is required for IL-33 signaling and activation of ILC2s, but then it's further required for the effects of effector cytokines, the type 2 cytokines, which drive the development of allergic airway disease. However, where we stand now, the mechanisms by which all these steps occur and how RAGE is involved is still unclear, and it requires further research so we can know the true therapeutic potential of RAGE. Is RAGE really a potential therapeutic strategy in obstructive lung disease? Well, let's look at what we know. We know that RAGE expression is highest in the lungs, and we know that it's increased in COPD and emphysema. However, studies are needed to know the expression of lung and RAGE in the tissue of those with asthma. We know that RAGE ligands are increased in asthma as well as COPD and emphysema. And on the side of eosinophilic and type 2 inflammation, I think it's been pretty well demonstrated that RAGE plays a critical role in mediating type 2 inflammation as well as mucus production. On the other side, we know a bit less, but we do know that RAGE is required for pollution and cigarette smoke-induced neutrophilic inflammation and mucus production, but we know less about the role of bacteria and viruses. We also know that RAGE ligands are increased and sRAGE decreased in neutrophilic airway disease. And it's very curious because we don't know the role of RAGE in neutrophilic asthma. However, we're currently investigating that in experimental models, and I think we'll have some very exciting data to share with everyone next year. But if this is the case, I think that this is RAGE has huge therapeutic potential to block inflammation, whether it's neutrophilic or eosinophilic, and also block mucus production and ultimately airflow obstruction. But there are things we need to consider. The role of RAGE in infection, much like with the use of corticosteroids, you can certainly reduce the inflammation, but do you make the subject more prone to infection? The role of RAGE in infection has been studied a bit, but not nearly enough. But in most cases, it shows that RAGE knockout mice do have better outcomes after lung infections. On the other hand, we have to think about the role of RAGE in lung development and aging, and we have to point out that RAGE knockout mice do develop spontaneous features of fibrosis in the lungs with extensive age. However, it's not clear that that is due to lack of RAGE or due to some other inherent error. Further research will need to know what would be the effects of, would there be negative effects of blocking RAGE over time. And lastly, there are a number of inconsistencies regarding the RAGE gene and its polymorphisms and whether it could be protective, causative, or if it has anything to do with the development of disease. So to summarize briefly, we know that RAGE signaling is dysregulated in COPD, emphysema, as well as asthma, with increased ligands and decreased sRAGE. We know that RAGE is a key mediator of persistent inflammation and mucus production and airflow obstruction in experimental models of COPD and asthma. And as we showed, that RAGE was critical at multiple steps during the development of allergic airway disease, but this may need to be looked at further in COPD and emphysema. But overall, I think targeting RAGE may represent a novel therapeutic strategy to reduce airway obstruction by inhibiting inflammation, mucus production, airway hyperresponsiveness, and obstructive lung diseases. So with that, I will say thank you to everyone for being here today. Thank you again to the organizers and the moderators, and thank you to my mentor, Dr. Ori, as well, for the opportunity to be here today.
Does RAGE play a role in IPF and ILD?
It's a little all over the place because in experimental models of fibrosis, RAGE knockout mice are protected from bleomycin-induced fibrosis. However, in response to asbestos, they have much worse disease. In silica, they have kind of just a different type of fibrosis develops with normal levels of fibrosis. In terms of IPF, it's very interesting to think, but it's hard to tell because sRAGE, like similar with COPD and emphysema, sRAGE levels are consistently decreased and associated with worse disease outcome. But it's unclear whether that is due to increased RAGE signaling or it's just an effect of destruction of the alveolar epithelium.
Is there any sex difference for RAGE expression and function?
That I'm not sure of. I don't know of any sex differences in terms of RAGE's function or expression levels. I don't see any different expression levels in the lungs in comparing female or male mice, for instance, but in humans, I'm not sure.
In the lavage fluid, bronchoalveolar lavage, can you measure sRAGE in there, and that could be a biomarker for inflammation and or for the disease?
I think so, yes. Certainly one of the slides I showed from the study in 2012 showed that S-RAGE was very much decreased and associated with increased neutrophils in the lungs of those with COPD and emphysema.
One of the questions in the chat, and I had that question too, probably both for Dr. Clunan and for Paul, that how the cells decide which way to die, or do you know that?
I think it has to do with a number of things. Probably the type of stress and the type of triggers that determines whether a cell goes into one or another pathway. Probably the whole environmental conditions like stimulus, pH, and everything that determines it. Maybe if you have something to add to that, Suzanne.
Yeah, I think, again, going back to the cell origin, take, for example, a type 2 alveolar epithelial cell, for example. We know that those cells have a lot more lipids floating around inside, right, because of surfactant and everything. And so perhaps it's a situation where they then become more primed or susceptible to any sort of lipid oxidation or lipid-mediated cell death. So that's just a simple example. I think some of the other cell types might have higher levels of other molecules that then make them primed towards certain stimuli. And we don't know anything about that yet, but I think that's what I would say. There's an underlying susceptibility to certain types of cell death.
What about former smokers? I mean, everything that we looked at that in cigarette smoking model or smoke extract in the cell line, BIS2B or primary cells, about 8% of former smokers develop COPD. So are these irreversible change?
I think it's well known that inflammation, I think alveolar macrophages hang around for a long time in former smokers. And also there's evidence in the alveolar type 2 epithelial cell for some long-term changes. So I don't see why not. That's my opinion. If the other speakers have any insight.
There is a comment in the chat window. In terms of soluble rays in BAL, there is good data that soluble rays remarkably increase in BAL in response to acute lung injury, like from hypervolipia and LPS. Any additional comment from the speakers?
I wonder if that has to do with just the damage to the epithelial cells causing shedding, or increases in inflammation and increases in MMP9, for instance.
I'm curious how this known cell death mechanism in COPD and in response to cigarette smoking, conventional cigarette smoking, could be applicable to e-cigarette smoking.
We have never looked into e-cigarettes, but I think as long as it's causing extensive damage to the epithelial layer, so if you have toxicants inducing damage, then you will have damp release and you will have a similar response.
Yeah.
Is it more like, so do you think is it more the proximal airway epithelial cell damage or alveolar epithelial cell damage? Because I'm asking this because from e-cigarette exposure, the most related pathophysiology is ARDS and EVALI. So that's more focused on the lung alveolar epithelial cells. And not many people actually look to it into large airway epithelial cells. So if either of you aware of that.
I think there's a strong lipid phenotype as well in those individuals as well. There's all these foamy macrophages and lipid accumulation. So to me, the alveolar epithelial cells, the alveoli in general would make the most sense in terms of being susceptible. But again, Simon says if there's injury there, you see it. But I think it's different from smoke-induced injury, right? It's very specific, and maybe that's down to the chemical nature of the chemicals in these vapes. But again, that might be quite interesting to tease out, because then you can say, well, okay, why is the alveolar type 2 cell more susceptible in vaping and not in smoke? Who knows?
Then again, bronchial epithelial cells are probably more exposed to this vaping, because most of the smoke will get to the bronchial part, but not to the alveolar part.
We also know that in cigarette smoke model or in human smokers that there is a decrease in clearance of apoptotic cells, and which is also pro-inflammatory. So can you comment on having apoptotic cells around, how that also interact with the cells that are dying via necrosis or necroptosis.
So I think when cells are unable to go into apoptosis, then there's a reason that they wanted to go in apoptosis in the first place. They probably have damage. So if they're unable to be phagocytosed as apoptotic cells, then ultimately they will find some other way to die. They will either go into necroptosis, or in the end they will be necrotic. So the defect in efferocytosis, so in the efficient removal of apoptotic cells, will in the end probably result in DAM protease anyway.
I don't know if anyone has anyone shown that alveolar macrophages and COPD are deficient in clearing necroptotic damaged cells. It's all apoptotic stuff, right? So I don't think anyone's shown that an alveolar macrophage cannot in COPD or smokers cannot clear a necroptotic cell. Actually, can an alveolar macrophage clear a necroptotic cell in general anyway? I don't know.
I don't know.
I don't think anyone's shown that. I mean, we're presuming that they do, right? But maybe not.
Yeah. So a question for team: that what's the future of this RAGE inhibitor? Do you have a plan? Where do you go next?
Yes, of course. We need to. I mean, the data we showed and we've published was just using direct type 2 cytokine, stimulation. But I think what we need to do is, first of all, see if it is also able to block everything in allergen-induced models, and really do it in an animal that has established disease, and then administer later to see if it really has a therapeutic potential. So that's some of the stuff we're working on now.
Is there any cell types that exhibit resistance to cell death in response to cigarette smoking, which might contribute a remodeling?
Yeah, I'm not completely sure if there are cells that are really resistant. If you give enough smoke, any cell will die probably, but we see large differences even if you compare different cell lines of bronchoepithelial cells, or if you compare that to cells of different donors, they will respond differently to cigarette smoke extract, and they will have a different response. So there's a lot of variability there. And also, considering different cell types, fibroblasts are a bit more resistant towards smoke, while epithelial cells are a bit more susceptible. So yeah, there is difference, but if you give enough smoke, any cell type will die.
I agree.
Do we know whether these DAMs actually cause barrier dysfunction?
I mean, based off other people's work, you could infer that they do, but no one has looked at it an ALI or, an organoid model or anything like that. We don't know specifically in the lung, which is an open area of investigation for a lot of people, I think.
Yeah, that's one of the recent the review published in Nature is about epithelial barrier hypothesis. Probably some of you know that. So that's related to increase in allergic and autoimmune disease and also other chronic conditions. So I thought it was interesting. Suzanne mentioned that kidney injury, and then you can detect the kidney injury marker. Is it supposed to be KIM, right?
Hmm.
The kidney injury marker was a KIM, KIM.
Yeah.
So but it was interesting that you can detect the mitochondrial DNA in urine and serum, but not in BAL. So I was wondering if that was a systemic effect, or the mitochondria really are released from the lung epithelial cells.
I think it's probably systemic. I know, Simon, you were able to detect mitochondrial DNA in the bowel for the mice, right?
Yeah, and also in humans, we were able to detect it, but we find far higher levels when you look at epithelial lining fluid, because it's a bit more concentrated in a local response. So that may, if you have access to the samples, I don't know, but then epithelial lining fluid will probably be a better source compared to BAL, which is of course very diluted sample.
Yeah. Also, the Spiromics cohort is a large. The processing may be different in terms of how they process their bowel fluid compared to what we would do in the lab, or, if you filter the mucus and things like that, you might remove the mitochondrial DNA. So it may just be a technical thing. But I think, systemically we know smoke has an effect on multiple organ systems, and it could be endothelial driven, as well. We know that endothelial cells are dysfunctional in COPD and in response to smoke. So yes, the barrier integrity may be compromised everywhere.
The question from Roma, Jeremy, it's intriguing that RAGE inhibition does affect IgE production. It will be interesting to see what effect this AHS on allergen-induced responses as indicated.
Yes, it's very interesting, especially the chronic house dust mite exposure. I'll point out as well, though, the same study that was in 2014 looking at Th2 cell sensitization, they showed that after house dust mite exposure, the mice had decreased levels of IgE, which is kind of the opposite of what we said. But I think part of that may be due to, I'm not sure, the model they used was more acute, and they also sampled their animals 3 hours after their last challenge, and we always do it 24 hours post the last one. But it's very interesting that the knockout mice after chronic exposure, they're making it seems like they should be having some sort of allergic response as they're making the IgE, and I imagine after the mast cells must be doing something there, but for some reason they're not. So it's very confusing, but very intriguing.
Okay, thanks everyone. I think it's been very exciting discussions all together with the three speakers, and of course the talks from three speakers been very stimulus and informative, and we really appreciate all of the time for taking the time to prepare these wonderful talks. So it's time to call off the meeting and symposium now. So thanks to everyone for joining the symposium as well. Hope you enjoy the rest of ATS meeting.